So in lab, we're going to use the wave nature of light to our advantage. Okay, so we're going to do an experiment to measure the concentration of food dye in a sports beverage. We're going to do that using a strategy called spectrophotometry. Okay, so spectrophotometry means exactly what it sounds like if you speak Latin, which probably we mostly don't. So spectro refers to the spectrum of light, essentially. So the spectrum of light is what I just showed in the first video introducing chapter 6. Photo means light, and metry means measure, like metric. Okay. So essentially, when we do spectrophotometry, we are usually using light to measure something. Frequently, that something is the concentration of a molecule that absorbs light. Okay, and so dyes, by their nature, absorb light. And so just to illustrate this idea, a leaf looks green to us, not now, it's, orange and red right now, but in the summertime, leaves look green to us because the entire spectrum, all the way from gamma to radio, so it includes all of the visible colors. The sun is a full spectrum light source. So the entire spectrum shines on the leaf and it absorbs everything except what you see. So it'll absorb all the purple, the blue, the yellow, the orange, and the red. And what we see is the color green, and that's called the transmitted light or reflected light. And the remainder of the colors are absorbed light, right? And so the symbol for that is just an A. We're going to use this phenomenon, okay? So if you have a sports beverage that is red, that means it's absorbing all the other colors of the visible light spectrum. So it would be absorbing orange, yellow, green, blue, and purple. If you remember your color wheel from art class, the opposite color is the most absorbed. So the opposite of red is going to be blue. So we would expect that your red sports drink is going to mostly absorb blue light. It will, of course, absorb some of the others around it as well, but mostly the highest amount of absorbance is going to be in the blue region of the visible light. And that's why we see red. So plants work the same way, and different plants absorb different pigments, use different pigments to absorb light. But a standard green leaf is primarily absorbing in the blue and the red, both two regions. You get two peaks, and I'll show you what that spectrum looks like in a second. So we have a few, this is actually technically called the Beer Lambert Law. Okay, it was invented or discovered by two different gentlemen, and essentially it relates absorbance to concentration. And you can actually use concentration in a variety of units. In our case in lab, we're going to use micrograms per milliliter because that's what your stock solution is made with. Dyes are very, you can use very, very low concentrations of dyes to get really bright colors. And one of the limitations of spectrophotometry is you can't have too concentrated of a solution or it isn't linear, which means your graph won't help you. So we use very diluted solutions, which is why we use micrograms. You should remember there are 10 to the 6 micro in the base unit. So if we're talking about grams, that would be in 1 gram. So you should remember this conversion factor from when we talk about the metric system at the beginning of the semester. You could also use molarity here. That's very common. And when you do, you would call this variable, the symbol is epsilon. It's a Greek letter. You would call epsilon molar absorptivity. In our case, we are not using molarity, so it is just called an extinction coefficient. This is only attained by measuring it in your exact solution. So even another student's results are going to vary compared to yours. So it's affected by all kinds of different variables like concentration, the solvent that's present, any contamination in the solvent can affect your extinction coefficient. So we just measure this every time we do a spectrophotometry experiment. You measure the background, and you're going to find that the slope of your line will be related to the extinction coefficient. And then, of course, absorbance is what we're actually measuring. B is the path length that the light travels through. And we control this by using a cuvette that is exactly 1 centimeter across. So B always equals 1. Okay. And so absorbance doesn't have units. It's kind of like a decimal value of a fraction, right? So it's really about the light coming out versus the light going in. So that's a ratio. So it's just a decimal value. It should always range between 0 and 1. If you're above 1, we call that outside of the linear range. If you're below 0, that's impossible. It can't absorb negative. It doesn't work. If that happens to you, then that means something in your calibration setup was incorrect, and you've got to fix that. Go recalibrate it. Okay, so it should always have absorbances between 0 and 1. Okay, so these are the kind of units we're thinking about. You need to do the reading assignment. This is also included in your syllabus and in the weekly week 9 assignment, but this is an important one so that you have the background knowledge to do the lab. So in lab, the first thing you're going to do is measure an absorbance curve. I have picked one from measuring the wavelength of different leaves. Okay, so on the every absorbance curve always has absorbance on the y-axis, and you'll notice it goes between 0 and 1, just like I talked about. And then the x-axis of an absorbance curve is always wavelength. In this case, we are going to confine our study to just the visible colors because that's what is mostly absorbed by dyes. There are other kinds of materials that absorb in the UV range or the IR range or what have you, but we're just going to look at visible light for now. So leaves actually have 2 different kinds of pigments. They have chlorophyll, which, as I said, since it's green, it's not going to absorb anything in the green area that's being transmitted to your eye, but it does absorb heavily in the blue region and in the red region. The other pigment in most leaves are called carotenoids. These are things like beta-carotene that's in carrots, lycopene that's in tomatoes and watermelon, astaxanthin, which is in anything purple. All kinds of fun names for these guys. So carotenoids tend to absorb, depending which one you're talking about, like lycopene looks red, so it absorbs more blue than the others do. But as a collection of different pigments together, carotenoid curves absorb in the blue, indigo. Now, there's some debate in physics circles about whether the color indigo is a legitimate color. I'm just going to call it indigo because I think it's less confusing than light blue. Okay, so this kind of greenish bluish color we'll call indigo. So yeah, carotenoids absorb in the blue, the indigo, and the green region. So leaves are actually able to harness quite a lot of the visible light by having different pigments in them to do so. In the fall, what we see happen is chlorophylls are temperature sensitive. So once it starts to get chilly out, they die. And when it stops absorbing in the blue and the red region, primarily in the red, right? Because carotenoids don't absorb there at all. What you'll see, the blue still gets absorbed by the carotenoids, but the yellows, oranges, and reds become visible. They're there all the time, and we just don't see them. The chlorophyll covers them, essentially. So in the fall, when the chlorophylls die because it's cold, we're going to see just the color of the carotenoids coming through. Okay. You can quantify how much carotenoid is in a leaf by crushing it up and dissolving it and then measuring the absorbance curve and then using some standards to compare it with. Okay, so the next kind of curve we're going to work on is called a standardization curve, and we have done these before. In this case, you have to make your dilutions. You're going to use volumetric glassware to do that. So that means we're going to always end up with 2 decimal places. So these should properly be shown like this, on and on like that. And you're always going to have a blank sample, which is no dye, just the water. You're going to use deionized water for this. We don't want to have any contamination if we can avoid it. And then you're going to make what we call a dilution scheme. All right, and so this is part of your pre-lab. You need to calculate this before you get in lab, or you're not going to be able to do the experiment. We're going to have 2 dyes, so you're going to have 2 different calculations because they start out at different concentrations. Don't forget that when you're doing a dilution calculation, you can use this equation, C1V1 equals C2V2. The C's in our case are going to be in micrograms per milliliter, and the volumes are going to be milliliters. And in all cases, you're going to always end up with 10 milliliters as your final volume. So you just need to solve for what your diluted concentration will be for all 10 samples. In order to find your initial concentration, this is going to come from the pre-lab. Okay, so we're going to use really careful glassware to do this. But, oh, interrupted myself. On your pre-lab, it gives you the concentration of the dye in the stock solution. There's 2 different ones. There's a red concentration and a blue concentration. The very first thing you have to do in order to plug it into this equation is convert first. So first you're going to convert it so that the units are micrograms on top and milliliters on the bottom. Make sure that you get rid of any number, so it should be per 1. PowerPoint doesn't want me to write there. It should be per 1 milliliter on the bottom, and however many micrograms you calculate on the top. Okay, so you're going to look at the pre-lab. You're going to do the unit conversion showing your dimensional analysis. Okay, so if you don't show your dimensional analysis, you're not going to get credit for that question. And then once you have it in micrograms per 1 milliliter, you'll plug that in as your C1. Your V1 comes from the procedure, right? So if it says to add 1 milliliter of stock solution, then you would put 1.00 here. Okay. V2 is always 10.00 milliliters, and so you're just solving for your C2 in that case. You're going to end up with, for each dye, concentrations from 0 stock added, which is just water. You don't need to use this equation for that, so don't waste your time. It's just going to be whatever you calculate in your unit conversions. All the way up to so 0 water added is what I'm talking about there. All the way up to all water, all 10 milliliters of water. You're going to make your solutions in test tubes. You're only doing red or blue in the lab. You're not doing both of them. But you're going to make them in test tubes using volumetric glassware like pipettes and burettes. And make sure you mix them up real well before you actually use them. And in the end, what you're going to produce is a graph that should be very, very linear. So you can tell the concentration of a dye by looking at it. The darker it is, the more concentrated it is. And you know this from everyday life, right? When you make Kool-Aid, if you put too much water in, you can see the difference. Okay, and so we're going to have a graph that's going to have absorbance on the y-axis and the concentration. In our case, we are going to be using the micrograms per milliliter unit, but the concentration will be on the x-axis, and it should be a very, very linear line. So lines follow the equation y equals mx plus b. In this case, our y is defined as the absorbance, because that's what we put on the y-axis. And our x is defined as the concentration. As long as our path length is 1, Beer's law says that the slope will be equal to our extinction coefficient. Okay, so m is equal to our extinction coefficient. Ideally, your intercept should be 0. This is assuming that everything is measured carefully and your DI water doesn't have any contamination, and also that your calibration for your blank is accurate. So in the next video, I'm going to show you how to do those things so you're prepared for lab.